Hi, thank you for listening to my talk. My name is Rebecca Westwick, and I'm a PhD candidate at the University of Kentucky studying under Dr. Clare Rittschof. And today, I'm going to be talking a little bit about how social signals and hive-level aggression interact to alter larval caretaking behaviors in the honeybee. Honeybees live in hives, which are large, organized societies. And within these societies, communication is critical for organizing the various tasks that need to be done. But because the brain can only process so much information at once, communication comes with trade-offs. Think about how much harder it is to have a conversation in a noisy, crowded restaurant than it is in, say, a quiet office. Instead of trying to hear someone in a crowded restaurant, honeybees rely in large part on pheromone communication. Worker bees within the hive will specialize on a small set of particular tasks at any given point. Bees with different jobs respond to particular signals that are pertinent to their line of work. For example, guard and soldier bees respond strongly to alarm pheromone, which coordinates aggressive nest defense behaviors. Nurse bees, who check on and feed the colony's brood, have recently demonstrated sensitivity to a different odorant, a chemical called e-β-ocimene that is released by larvae in response to starvation. Now, nurse bees generally won't become aggressive in response to alarm pheromone the way that guards and soldiers will, at least in lab tests. The nurse bee's job isn't to defend the nest, so they don't react aggressively when presented with this signal. But just like being in a crowded restaurant, nurse bees that are trying to pay attention to their e-β-ocimene signal in a hive setting are constantly swimming in a soup of unimportant information, such as alarm pheromone and the dozen or more other pheromones that are found in a hive. So that leads to a question. Do these conflicting social signals alter the ability of nurse bees to focus on larval signals and respond appropriately? We chose to focus on aggressive social signals as the potential conflicting information for this study because hive-level aggression has previously been shown to affect the adult behavior of the larvae that were raised inside of that hive, possibly through the actions of the nurse bees that raised those larvae. To assess this question, I performed a rather complex nested experiment that tried to capture all of these variables together. Over the summers of 2019 and 2020, I took nurses from hives that were known to be either high or low aggression and put them in an observation hive under red light conditions. I would take a frame of honeycomb that had been sourced from another hive, and I would mark cells that contained either a larva to which I added extra e-β-ocimene, or were empty. Then I would apply either alarm pheromone or mineral oil as a solvent control, and then video record the nurses for 10 minutes. Then, with all of that video data, an individual who was blinded to the treatments would count how many times each marked cell was visited by nurses in that 10-minute period, as well as the latency to the first visit. So to clarify, the treatments that we had in this experiment were the aggression level of the hive the nurses came from, either high or low, the alarm pheromone treatment, which was either alarm pheromone or mineral oil as a control, and the cell type, which was either a larva with added e-β-ocimene, a larva alone, or an empty cell. The e-β-ocimene was applied in a way that it would be a more persistent signal, whereas the alarm pheromone treatment was a brief, one-time application at the beginning of the trial, mimicking a single predator event. To get a sense of what our scorers were looking for in terms of a nurse visit, you can see that we have a nurse bee here, and she dunks her head and maybe some of her body into the cell to check on the larva that's at the bottom. Our first result was that e-β-ocimene was not doing what we thought it would. For the graph, the x-axis shows the cell treatments, either a larva with added e-β-ocimene, a larva alone, or empty, and the y-axis is the mean number of visits with standard error bars. Since a previous study showed in a laboratory setting that e-β-ocimene was released by starved larvae and increased nurse visitation, we expected that it would increase nurse visitation to the larvae we applied it to in our experiment as well. Instead, we found that adding e-β-ocimene to a larval cell actually decreased visits fairly drastically, down to about the rate of visitation to empty cells. We're currently investigating why this might be the case. However, for the purposes of today, we will be excluding the e-β-ocimene data from all further results since we're not confident about what's happening there. On to the main result addressing our central question. This graph is similar to the one in the previous slide with cell type, either larva or empty, on the x-axis and mean number of visits on the y-axis. This time, though, we've broken out alarm pheromone in red and mineral oil in blue. And on this first graph, we are only looking at nurses from high aggression hives. You can see that these nurses from high aggression hives visited both empty cells and cells with larvae less frequently after alarm pheromone was applied, suggesting that the nurses may indeed be getting distracted by this irrelevant social signal. On the other hand, nurse bees from low aggression hives did not decrease their visits in the presence of alarm pheromone. These results suggest that nurse bees originating from hives of different aggression levels are changing their parental care behavior in different ways when a conflicting social signal is applied. And indeed, I ran a generalized linear mixed model with all of our main factors as fixed effects, which would be hive aggression, alarm pheromone treatment, cell type, and their interactions. And I also included hive ID and treatment order as random effects. When I ran the model, the interaction between hive aggression and alarm pheromone was highly significant, reinforcing our interpretation that nurses from hives of different aggression levels show opposing responses to alarm pheromone. This experiment has shown that social signals can influence parental care behaviors in honeybees. Additionally, the way these transient social signals modify behavior is dependent on the hive-level social and aggressive context. It has been well established that social cohesion within a hive is critical for both individual and hive-level health. One prominent example of this is how honeybees, like many other social insects, rely heavily on social immunity to ward off disease within the hive. The research I've presented today demonstrates a potential mechanism by which social interactions could have reverberating effects on the next generation of worker bees, since the actions of the nurses actually play out their effects on the larvae. And with that, I would like to thank you for your attention and your time. I would also like to acknowledge everyone listed on this slide. If you have any questions, please don't hesitate to reach out to me.